Okay. So we have 6 participants. 8. We might wait for 30 seconds for more participants. Yeah, I will start my presentation now. So hello everyone, it's a great pleasure to give you this online presentation on Germ-Free mice for microbiome research and drug development. This presentation will be given in 3 parts. I would like to kick off with an introduction of Germ-Free mice, then discuss their application, and finally introduce our platforms and service. Let me introduce some background information of Germ-Free mice first. Also, Germ-Free animals have been studied over 100 years. Not many people know what are germ-free animals, or more specifically, what are germ-free mice. The term germ-free refers to an animal being completely devoid of microbes, including bacteria, virus, fungi, parasites, and protozoa throughout its lifetime. The concept of germ-free animals was put forward more than a century ago by Liyou in 1885. At that time, he believed that surviving without bacteria was impossible. However, 10 years later in 1895, Nuttall and Tefilder at Berlin University produced the first GF animal, which is junior pigs, survived for 13 days. Then Kuster designed the first isolated prototype in which the germ-free goats were maintained for months. Till 1946, the first germ-free rat was established. And in 1950s, isolators were invented, which is light, practical, inexpensive house for germ-free mice. So after the house was built, the first GF mice were successfully developed in 1959. So according to the definition of national standard and the microbial containment, experiment animals can be divided into clean grade, SPF grade, gonotobiotics grade, and germ-free grade. The clean mice was maintained with minimal biosecurity and health surveillance and not screened for any microbes, including pathogens. Specific pathogen-free mice were partially screened for microbes and free of specific pathogen. Gonotobiotic mice refer to mice in which every microorganism present is defined. Same with germ-free mice. Gonotobiotic mice were also raised and maintained in germ-free isolators. And as mentioned before, germ-free mice are free of all microorganisms. So nowadays, SPF and GF mice were extensively used for scientific researches. So comparing with SPF mice, some differences were found, mainly in the anatomical and physiological characters, such as GI tract, circulatory system, immune system, metabolism, and reproduction system. So let's dive into this one by one. You can see here the most significant change of germ-free mice is their gastrointestinal tract, of which has an enlarged cecum due to the accumulation of undigested food debris. It has been reported that the intestinal wall and mucosal layer of germ-free mice are thinner, and the removal of intestinal epithelium is slower than that of SPF mice. So for the circulatory system, germ-free mice have smaller heart, lower cardiac output, reduced blood volume, and increased RBC count and hematocrit output. In addition, the immune system between germ-free and SPF mice is also different. Specifically, germ-free mice are deficient in lymph node tissues, including spleen, thymus, and lymph nodes, leading to a decreased CD4 plus and CD8 T cells. In addition, the gut-associated lymph node structure are also influenced by the absence of microbiota, so making germ-free mice has a deficient gut. Furthermore, the number of IgA expression B cells is decreased, resulting in the reduced production of IgA due to these mentioned immune deficiencies. So GF mice are more suspicious to infections than SPF mice. So another one for the metabolic system, Germ-Free mice are characterized with decreased body fat and decreased metabolic rate, but increased food and water intake. So next to that, the absorption and retention of calcium and magnesium in GF mice is better than that of SPF mice. Last, GF mice are more sensitive to high cholesterol diet, and it has been reported that the level of blood cholesterol in GF mice were 2 times higher than that in SPF mice when fed with high cholesterol diet. So for the reproduction characteristics, as the absence of gut microbiota can affect their reproduction, Germ-Free female has prolonged the estrous period, reduced the estrous frequency and copulation, and reduced the implantation and fertility rates. So what you see here is some of our data showing the anatomical and physiological differences between SPF and GF mice. The result shows that SPF mice have a higher body weight and bigger heart, lung, liver, spleen, and penis than that in GF mice. But the cecum of GF mice is twice bigger than that of SPF mice. So our own data recapitulates most of the phenotype differences that are reported in literature. But interestingly, also the differences were found in the physiological characters between SPF and GF mice, hematological and blood chemistry, but the biochemical parameters had no significant differences between Germ-Free and SPF mice. So this is our first part. So for the second part, I would like to introduce applications of GF mice. Briefly, the application of GF mice mainly in 2 aspects, so which are interaction and selection and evaluation. For the interaction part, GF mice can be used for comparing the physiological differences between them, which has we did before, and like what I just discussed. So second, it is a great model for investigating the function of certain genes, because this model, Germ-Free mice model, eliminates the microbial interference. So third, they can be used for studying the cause effect, that is, whether microbiota is associated with or leads to certain diseases. So for that, GF models allow us to explore the impact of gut microbiota on disease development of different aged mice. So for the selection and evaluation part, GF mice can be used to explore live biotherapeutic products by colonization of LBP candidate in GF mice models. In addition to that, GF mice can be used for construction of humanized gut microbiota mice model, which allows for the evaluation of functional food and therapeutic agents. So these are all applications, or let's say potential applications of Germ-Free mice. Now we understand that Germ-Free mice can be used for this type of researches, but which research area or which researchers now are using GF mice? Let's see. So first is oncology. With the pop-up of 2 clinical studies reporting that fecal microbiota transplantation could enhance the response of PD-1 antibody in non-response melanoma patient, researchers about gut microbiota and anti-tumor agents interaction were booming recently. So the following 2 papers study the anti-tumor effect, or let's say the effect of microbiota on anti-tumor agent by using GF mice models. So this is for the oncology part. In addition to that, the immunological research also use GF mice. Gut microbiota can affect our immune response, I think you all know. And in order to study the underlying mechanisms, GF mice is indispensable models for those interactions. So next, for metabolic diseases, researchers also require Germ-Free mice models, as shown in this paper. Researchers transplanted the microbiota from twins discount of obesity patients into separate groups of recipient gene-free mice. The fecal microbiota from obese twin can lead to an increase in body weight with the low-fat diet, while transplantation of the lean co-twins microbiota did not show this effect. Here, the gene-free mice is a good model to study the cause-effect relationship between gut microbiota and diseases. So for the neurological researchers, gut microbiota can affect our neural system via the gut-brain axis. This paper demonstrates that gut microbiota enhanced the motor defects, microglial activation, and alpha-synuclein pathology. They also found that after colonization of the microbiota derived from Parkinson's patient into gene-free mice, the motor dysfunction was enhanced. So to sum up, gene-free models were used in many research areas nowadays, and the usage of gene-free mice in microbiome researches definitely can add the credibilities of our research paper. So this is the second part for the applications. So the last part is about our platforms and the service that we can offer. So our platforms were made up by two branches. The first is gene-free mouse models supply platforms, which is a place where we raise and maintain our gene-free mice. The second is research platforms, where we have our own microbiology lab, animal experiment platform, and immunology lab. The research platforms allow us to provide high qualified experimental service for our customers. So let me first introduce our gene-free mouse model supply platform. So the mouse supply platform has nearly 2,000 square meters, containing GF animal facilities and hundreds of GF isolators. 3 bedding rooms contains 1,300 cages were used simultaneously to ensure enough yield. To make sure the gene-free status of our mice, we use the standard protocol for material sterilization, including autoclave, peroxy acid disinfection, and irradiation. For each batch of material, biological indicators were used to ensure our sterilization process is effective. So what you can see here is these are the current available GF mice models in stock, so including the normal inbred strain GF Black 6, RBC, outbreed strain ICR, and other disease or immune deficient strains such as NCHX, or we also have IL-10 knockout mice, gene-free mice. So after you buy these GF models from us, we will deliver them to you. So especially for the overseas customers, after you purchase some models, we'll use shipper sleeves to transport. So the maximum capacity of the shipper sleeve are 2 cages. Each cage can contain 6 to 8 mice, depending on the cage. As their age, for 3 to 4 weeks mice, 60 mice can be put into the shipper sleeve, and 40 mice for 5 to 6 weeks old, and 12 mice for 7 to 8 weeks old. So according to our experience, this kind of delivery manner has been proved to be effective to keep a sterilized environment during the transportation. So in addition to the GF mice cells, in our gene-free supply platform, we are also able to do the aseptic purification, which was achieved by either in vitro fertilization or case section. As we proceed with our knockout ore project, we already obtained more than 20,000 conditional knockout mice. So combining the Pope KOAP mouse with our aseptic purification technique, I guess you can already imagine how many kinds of gene-free mice we can get. So this is also why I call it Mission Possible. So let me summarize a bit. So our gene-free mouse supply platform can of our surveys include first, different germ-free mice strain cells, and second, aseptic purification. This is our first platform. So next to that, we have our research platforms. The research platform was consist of microbiology lab, animal experimental platform, and immunology lab, which allows us to provide one-stop experimental service for our customers. So for the microbiology lab, we offer service like bacteria identification and preservation, bacteria quantification and quantification, and fecal-derived microbial extraction, and also anaerobic culture. So for the animal experimental platforms, what you can see here is a workflow of our platform. So everything before starting animal experiments will be sterilized, as mentioned before. Followed to that, all the needed materials and germ-free mice for experiment will be put into isolators, and all the experimental execution will be carried out in isolator. Alternative to isolators, for some projects, we also use iso cages and perform experiments in the coupled biosafety flow cabinet. So after the termination of animal experiments, we have immunology labs that allows us to do multiple analysis. We got cell culture lab enable us to do the cell-related analysis, and flow cytometer to do cell sorting, which is needed if you want to study the microbiota and immune system interaction. And lastly, histological lab for staining, immunohistochemical analysis, ELISA, Western blot, and general blood chemistry, and also hematological analysis. However, what service we can offer based on those research platforms? Actually, we have already done hundreds of projects from both academia and industry customers. These projects include surveys or techniques like fecal microbiota transplantation, microbial colonization, drug efficiency evaluation, disease mode construction, and also some other customized service. I would like to elaborate this service in the following slides. So for the fecal microbiota transplantation, here is a brief description. So no matter if it's the feces is from mice or humans, the first step is extraction. So we will first extract the microbiota isolates from the feces, and then we will transplant them into germ-free mice bag garbage. Normally, feces will be collected during the experiments for bacterial detection or sequencing, depending on the design of specific experiments. So in the end, the customers normally will perform 16S sequencing on collected fecal samples to compare the similarities of the flora before and after transplantation to evaluate the transplantation efficiency. What I showed here is a project we conducted before, investigating the recovery of phenotypes after fecal microbiota transplantation. We can clearly observe that the secant was dramatically decreased and the liver was increased after fecal microbiota transplantation. So I guess now it is very clear how we perform the fecal microbiota transplantation experiment. So of course, next to that, single bacterial colonization was performed by us a lot. Comparing to the fecal microbiota transplantation, the process is relatively simple, but the germ-free environment maintenance is much more strict. In brief, bacterial strain was recovered by anaerobic culture, after which administered them to germ-free mice bag garbage. What we should note here is the frequency of single bacterial transplantation is really depends on the property of the colonized strain, like whether the strain is acid resistant or biosalt tolerant, whether it is easy to culture or colonize. So what you can see here is pterosid bacteria that we colonized and the verification of the bacteria. So this is for the microbiota and bacteria parts. So in addition to those two parts, we actually conducted an oncological study before, and this study was a basis for further anti-tumor studies. Specifically, we have inoculated a tumor cell line subcutaneously in both SPF and GF mice after the formation of the solid tumors. Anti-mouse PD-1 antibody was administered to mice, and the anti-tumor effect of antibody was evaluated in GF and SPF mice at the end. So the results showed that the anti-tumor effect of anti-mouse PD-1 is less efficient in GF mice, which might be due to the deficient in the adaptive immunity. But this also gives us a larger window to study the anti-tumor effect of a combined use of immune checkpoint inhibitors such as PD-1 with other probiotics. So adding to that, there is an ongoing oncological study aiming to evaluate the anti-tumor effect after the combination of certain bacteria with anti-mouse PD-1. So what I want to emphasize here is, even though our experiment used only anti-mouse PD-1 antibody, it does not limit it to this one certain immune checkpoint inhibitor. We can also perform other oncological studies that our customer want. So last but not least, we also have constructed a lot of disease models, which is also part of our customized services. So we have constructed many disease models like by different methods, of course. So including osteoporosis model by ovary removal, and obese and colon cancer model by high-fidelity induction, and pancreatitis by drug induction, and other disease models like we also impair the gut barrier integrity by hyperoxia. So this is what you see as right is a paper we sorry, this paper published we cooperate with our academic partners. So taken together, our platforms provide service includes but not limited to derivation of gene-free genetic engineered mice, in-stock gene-free mice supply, including black eggs, PBC, ICR, NCG, and ionoca streams, and fecal bacteria or single bacterial colonization, evaluation of the anti-tumor effect of bacteria, and GF disease model construction, and also other customized services. So if anything I present fits your research interest, or anything of our service can help your research, please do not contact us via this contact information. So thanks a lot for your attention, and all questions are welcomed. So for the question, I will read it out. If the sterile mice are put into sterile bags, can they be raised in SPF condition? So my answer is no, because if you want to keep the GF mice germ-free environment throughout its lifetime, or you want to use GF for any further experiments, you have to make sure that the before experiments or also throughout the environment throughout your experiments, they have to be completely germ-free. So any other questions? Let's see. Does my answer your question, Marvey? So what is the frequency commonly used for FMT? So this is actually a good question. So it's a practical question. Normally we'll do 3 times transplantation with 1 day interval, and duration is 1 week. So after 1 week of 3 times, normally the microbiota will be colonized. So just let's see. So what is your general method to detect the derivation test? So I think you mean what's the method we detect whether our Germ-Free mice was already be contaminated. So for the contamination detection, we normally have two methods. So first we can stain their feces and check under the microscope to see whether there are staining bacteria. This is the first method. So for the second method, we can do the anaerobic culture. And to after like 1 or 2 days, we can count, we'll see whether there's some bacteria on the agar plate. And the third method is PCR. I hope I answer your question, Minji. So the next question is GF mouse more sensitive to some tumor or metabolic disease? This is actually a very good question. So for Lu Chen, so for this question, because I think for even so for different tumors in SPF mice, they have different sensitivities. So you put different tumors to no matter it's GF mice or SPF mice, it will go differently. So there's no study compare whether I mean, let's say there's no study showing that GF mice is more sensitive to those things, to different tumors. So there's no comparisons. But definitely, if you use different tumors, you use different tumors to inoculate them, of course, they will grow differently. Is this your question, Zhang Lu Chen? So next question is, since there is such a big difference between sterile mice and SPF mice, can they grow normally for solid tumor? So if I understand you correctly, so you're asking whether the solid tumor can be formed in GF mice, right? So according to literatures and also our own data, of course, solid tumor can grow, let's say, can form a solid tumor in GF mice. But the size of the tumor is different because GF mice is immune deficient. Of course, have different. Let's say the size of the tumor formed in GF mice is different with in SPF mice. So, any more questions? Let's see. Yep. So actually, adding to your question, Mawei, I would say, according to our own data, we found the tumors formed in GF mice is smaller than that in SPF mice. So based on this, and also the antibody, let's say ICI, the immune checkpoint inhibitor, is less efficient in GF mice. So based on this fact, actually, if you want to study whether certain probiotics or certain microbiota, let's say bacteria, can enhance the effect, the anti-tumor effect of certain antibodies, this is, I would say GF model is great model because they give you a large window. Also, I see one more question. So compared to the antibiotic mice, what's the advantages of GF mice? Because yeah, this is a fact because it's actually very expensive. So for the compared to the antibiotics mice, so GF mice is completely devoid of any microbiomes. So if you want to study the effect of like one bacteria or the interaction between one bacteria or a few bacteria with your immune system or with other phenotypes, you have to use Germ-Free mice. So for the antibiotic treated mice still have some bacteria left, and it can also induce growth of the antibiotic resistant strains. So this is the very obvious advantages for the GF model. Thanks for your questions. So if there are no more questions, I would like to close this webinar. And thanks for your time. Thank you.